Hi, my name is Yuan, and in this video, we will go through my tutorial on integrative analysis of single-cell multiomics data using deep learning, and we will be running our tutorial on Saturn Cloud. If you have never heard of Saturn Cloud is a cloud platform for data scientists in the biotech field, supporting use cases including single-cell analysis, high-content screening, collaboration across teams, big data, and more. We will first start our Jupyter server. And this may take a few minutes. Now we can start our Jupyter lab. Now we open the GitHub repo for our project, and we start our Jupyter notebook. Single-cell sequencing technologies are being widely used in biomedical research. For example, researchers from around the world have been using single-cell technologies to profile and dissect human immune responses against COVID-19 and have made numerous important discoveries about the pathogenesis of the disease. In addition to single-cell RNA sequencing, which measures the transcriptome or gene expression of single cells, newer technologies such as cite-seq can measure multiple molecular modalities, including proteome and transcriptome from the same cells. As shown in this figure, by using oligoconjugated antibodies, cite-seq can provide sequencing-based readouts for the expression of hundreds of proteins along with gene expression. This allows us to obtain more comprehensive information about the biology of individual cells, but also requires new computational methods that can effectively integrate single-cell data from both modalities. In this tutorial, we will be conducting integrative analysis of cite-seq data using an unsupervised deep learning method called autoencoders. We start off by importing all the libraries that we would need, including some standard libraries, PyTorch and PyTorch Lightning for implementing the neural network. We use PyTorch Lightning to simplify the training process. Next, we import UMAP and Plotly for visualization purposes later. We also use TensorBoard to monitor the training process. We call the cite-everything function from PyTorch Lightning to make sure that our results are reproducible. We use GPU if there is one available, otherwise we would use CPU. Because the neural network in our example is not very complex and the dataset is not that big, CPU would be sufficient. Regarding the data, we would use the cite-seq dataset generated by this publication back in 2019, where the authors measured the gene expression of about 30,000 bone marrow cells together with the expression of 25 proteins. I have already pre-processed the data to generate normalized counts and the cell type annotations using Seurat, which is a very popular R package for single-cell genomics analysis. You can find the script here if you are interested, and there are 3 CSV files which we can download using this code if they are not already downloaded. Once we have the data ready, we can import them using pandas. We can verify that indeed there are about 30,000 cells and we have 2,000 genes along with 25 proteins. Next, we can concatenate RNA and protein data into a single pandas data frame, and we can verify that the new data frame has 2,025 columns, where each column is a gene or protein, and each row is a cell, and the data are normalized counts. We also import cell type annotations for visualization purposes, and we modify some of those annotations, and we also use the label encoder from scikit-learn to convert those cell type annotations that are in the string format to integers so that we can feed them into our neural network. Next, we define our PyTorch datasets and data loaders. We built a custom dataset for our tabular data. So we have the data for the cite-seq expression data, and the labels are the cell type annotations. We have to implement the lim function, which returns the total number of cells in our dataset, and the getItem function will return a single cell based on the given index. So the X would be the cite-seq expression data, and the Y would be the cell type annotation for that individual cell. Once we create this dataset, we can use the cite-seq data frame and the labels to generate our dataset, which is then split into train, validation, and test datasets using the random split function from PyTorch. In our case, we use 70% of the cells for training, 15% of the cells for validation, and the remaining 50% of the cells for testing. We can take a look at the number of cells for each set. We have about 20,000 cells for training and 40,000 cells for validation and testing. Once we have the data sets, we can build our data loaders. In our case, we choose a batch size of 256, meaning that we're gonna group 256 cells in a single batch and feed that into our neural network. Once that's done, we can take a look at one single example of our data set. You can see that the data would be the cite-seq expression data, and the label is the numeric or integer representation of the cell type annotation. Next, let's take a look at the autoencoder architecture. So it has two major components, a encoder and a decoder. The encoder would compress the input data into a much lower dimension, and the bottleneck layer would store the latent or compressed representations of the input. And then the decoder would try to reconstruct the input from the latent representations. And the goal is to make the reconstructed input as similar as possible to the original input. Some of the common tasks in single-cell analysis include dimensionality reduction and clustering. And since our autoencoder compressed the input data into a much lower dimension, we can use the latent representations for these common tasks. In our case, since we have 2,000 genes and 25 proteins, there is a dramatic difference in terms of the dimensions of these two data modalities. Therefore, we first encode them separately using two different encoders, and then we concatenate the outputs and then pass that through another encoder to generate the latent representations. And the decoder would try to reconstruct the input from the latent representations. We first define a module that groups linear, batch norm, and dropout layers as fully connected layers so that it would be easier to implement the encoder and the decoder later. Now we can implement our encoder, which, like I mentioned earlier, consists of an encoder for RNA, an encoder for protein, and a final encoder for the concatenated output. For the RNA, we use two fully connected layers to reduce RNA data into a given number of dimensions. For protein, we use only one layer, and then there is another fully connected layer for the final encoder. So in the forward function, we run the protein and RNA data through their corresponding encoders, and then use the final encoder to generate the outputs. The decoder is basically a flipped version of the encoder, and as we can see that We have three fully connected layers. It starts from the latent representations and then construct the input from the latent representations. And the output from the decoder would also be the final output of our autoencoder. Once we have the encoder and the decoder defined, we can assemble them into our autoencoder, which is defined as a PyTorch Lightning module to simplify the training process. We would first need to define init, where we save the parameters and create our model. As we can see that the model consists of an encoder and a decoder. In the forward function, we use this for inference. In our case, we use it to generate the latent representations of the input. We define the configure optimizers function to create a optimizer for our neural network. In our case, we use Atom. And we also define a learning rate scheduler so that the learning rate would decrease if there is no improvement on the validation set. Finally, we need to define the training step, validation step, and the test step. The training step would need to calculate and return the loss of a single batch. To this end, we define a helper function where we run a single batch through the encoder and the decoder and then compare the similarities between the reconstructed input and the original input using MSE or mean squared error loss. The validation step and the test step are very similar to the training step, except for that we apply this function to the validation and the test datasets. Once we have the autoencoder defined as a PyTorch Lightning module, we can use the trainer API from PyTorch Lightning to execute the training process. First, we group all the steps into a function to make our life easier, and we create a PyTorch Lightning trainer, and we use GPU if it is available, and we train at most 50 epochs. We use the model checkpoint callback to save the best performing model, and the learning rate monitor logs the learning rate as the model is training. Next, we create our model. In this case, we choose a dimensionality of 30 for the RNA data and a dimensionality of 18 for the protein data, and the final latent representation would have 24 dimensions. Once this is done, we call the fit function to train the model using the test data loader and validate on the validation data loader. After the training is done, we can test our model using the given data loaders using the test function. After all this is done, we return the trained model as well as the results. All right, I forgot to run this cell, and we run this cell, and now we can train our model. This may take a few minutes. Okay, once the training is done, we can take a look at the loss for the training, validation, and the test data sets. As we can see that they're very comparable, meaning that there is no overfitting. It is nice that PyTorch Lightning automatically logs the training results into TensorBoard, which we can open here. We can monitor the learning rates as well as test loss, training loss, and you can see that the training loss goes down or decreases over the epochs, and similarly the validation loss also decreases over time. Since our autoencoder compresses the input into 24-dimensional vectors, we can then project the representations into a 2-dimensional space using UMAP for visualization to see how the different immune cell types cluster in our latent space. We use our trained model to generate latent representations of our test data set, and then we also save the cell type annotations so that we can color the cells by their annotations. Next, we run UMAP, and then we can use Plotly to visualize the UMAP. As we can see that our autoencoder model does a pretty good job separating those different immune cell types as they form distinct clusters in the UMAP space. For example, here we can see our CD4 T cells. These are CD8 T cells, B cells, NK cells, progenitor cells, and the monocytes. We can also visualize the latent representations using TensorBoard. We just need to write the embeddings first, and then we can open a TensorBoard to look at or explore the representations. So this is PCA by default, and we can color the cells by their cell type annotations, and we can choose UMAP. And we can see that these different immune cell types form nice clusters in the UMAP space. For example, we have the monocytes, progenitor cells, CD4 T cells, B cells, and CD8 T cells, and NK cells. So in summary, in this tutorial, we implemented an autoencoder using PyTorch and PyTorch Lightning for integrating the protein and RNA data generated by cite-seq, and the latent representations generated by our autoencoder can then be used for downstream analysis, such as dimensionality reduction and clustering. I hope you enjoyed this video, and please let me know if you have any comments. Thank you.